Dear President, and thank you very much that you are here. And I will continue in Russian because the auditorium here is mostly Russian language speaking persons. Спасибо большое за приглашение.
Thanks a lot for the invitation to this Congress. It is a pleasure for me to share with you the results that we obtained when we used the protocol, which today is one of the most serious protocols for us, because traditionally we have always worked under the umbrella of the German protocol. Before I present our results, I would like to tell you that today we have a new classification of 2016, which has been already published. And on the 9th of May, an article was published highlighting new data on histomolecular features for some tumors. In particular, we know there are numerous variants of medulloblastoma, and they may look differently histologically under a microscope, that there are molecular variants of medulloblastoma, and there are more than 4 types of them, and they are very different in terms of their features, in terms of their prognosis, and in terms of our approaches to treat those medulloblastomas. Currently, we hope that we are to abandon the so-called grading system of CNS tumors, because glioblastomas of grade 4 and medulloblastoma are different in terms of their features and prognosis. As to ependymomas, I'm not going to speak of them because the previous speakers already described 9 groups of ependymomas, and we know that there are ependymomas of a favorable prognosis and unfavorable prognosis. As to atypical teratoid tumors that are included into the group of embryonal CNS tumors, there are also various subgroups, and most likely the future classification will incorporate these subvariants as well. And this is how the protocol looks like, which we currently use in our center. It is also used in other clinics of the Russian Federation. Then we also have a so-called registry, and we collect data on those patients who have been treated starting from 2012. Here you see the formal approval of the HIT-MED-Study Group, Professor Rudkovsky, allowing us to use and adapt this protocol in Russia. In 2015, this protocol was translated and adapted for our local conditions, and starting from 2012, we have already more than 500 patients in this registry that can be treated on this protocol. Here you see the registration form for the prospective study that we started in February 2015. This is the presentation of the logistics of the interaction with different sites after the surgical treatment and determination of the tumor type. Reference analysis is done in our center, and not just to have immunological verification, but histochemical verification, MRI, cytology of CNS, then stratification of patients follows, and treatment is agreed upon. This is how the reference data look like. We do it jointly together with our German colleagues, and thanks to the online system, we have a possibility to quickly get the results if we face some difficulties in our center. Why it's important? My colleagues who already spoke at this conference noted that it's very important for us to timely detect changes in MRI scans, and it's important to do this type of investigations in adequate conditions, and it's very important for us to detect changes that are taking place in the patient and interpret them correctly and modify our tactics of treatment based on them. The reference based on MRI is important, but also radiation therapy is also important too. Here you see metastatic lesions of the temporal lobe of medulloblastoma, and this patient was treated in an adult center. Regretfully, special shields were on the eyeball that actually shielded the zone of the brain. And in this area, there will be a typical problem. We also see it as their metastases develop. That's why the reference data at all stages is very important so that we could correctly interpret the results and make our correct decisions on treatment. Starting from 2015, we already included 38 patients into this study. Median age was 6 years, predominance of boys. As in all other studies on brain tumors, surgical interventions had an equal split between federal centers, primarily localized in Moscow, and in some clinics in the regions, and the regional clinics. That is, the split is 50 to 50. Histological type. We saw the predominance of medulloblastomas. Those were patients with M0 stage. MEC amplification was done in patients, in 11 patients, that is, in 50% with medulloblastoma, and one positive patient was found on MEC. And today, we have a chance to run molecular tests in case of expression of beta-catenin, and we had a positive case. It was a mutated CT1 gene. All patients were treated based on HIT-MED guideline. 2 had medulloblastomas. At the initial stage, high-dose radiation therapy was also used. 4 patients received hypofractionated radiation therapy and 27 intensive modulated radiation therapy. All patients are alive. 8 of them completed chemotherapy and radiation therapy with complete response. 1 patient had a recurrence with anaplastic ependymoma, and we believe that all positions of the protocol are quite feasible in the Russian Federation, and we did not see any adverse events, serious adverse events on such therapy. Therefore, we can say that modern standard approaches to diagnosis and treatment for children with high-grade neuroepithelial brain tumors, including molecular biological studies, are possible and feasible in Russia, and this makes it possible for us to take part in international studies and trials. I would like to thank all of our patients and their families who entrust their children to us. And I would like to thank all the maintenance personnel that work in our center and regional clinics and take active part in the care of children. And I would like to thank the charitable foundations and separately Professor Korshunov and Rumyantseva, as well as a group of neuro-oncologists that work in our center, radiology specialists, interventional radiologists, and regional doctors who are very helpful in the management of our patients. Thank you very much.
Yes, we are significantly overrun, so I don't think we have time for the discussions here. So, but I would certainly you agree with us that we have learned a lot from our speakers. So we have got a lot of insights in terms of scientific research, in terms of the future challenges. So with this, I'd like to close these sessions, and we will have to pass the stage to the next sessions. Thank you for your attention.